Namaste to all. Welcome to the new session on drug discovery or drug designing. So in this session, we are going to see about the next topic, that is about the drug target identification and validation. And in this particularly, we are going to see about the role of bioinformatics in the analysis of nucleic acid and protein sequences. So in our previous session, we have already seen that how the drug validation and identification is done by other tools, and how, like mass spectrophotometry, or you can say like LMS and other things. So LCMS and all, like liquid chromatography and mass spectrometry. So in that order, we are going to see about that how the bioinformatics plays a very vital role in the drug discovery. So if you get any sample, that is a query sample, you can put it in the query sample, and based on the database, you can analyze your sample, saying that whether any similarities are available or whether any possibilities are there or similarities are there for the particular drug aspect. You can also find it out using the protein sequences, that you can create a homology model by using the sequencing and the bioinformatics tools, by which if any domain or motif is available where your drug can bind, then that will be very much useful. So in this order, the basic thing what we are going to see is we are going to see about the drug discovery aspects. So in this, the basic thing is, okay, so the sequence analysis. So if you see this in bioinformatics, sequence analysis and the alignment plays a very vital role. So in sequence analysis means it is the process of subjecting a DNA, RNA, or peptide sequences to any of a wide range of analytical methods to understand its features, function, structure, or evolution. So we know that in this process, like if you are getting a DNA sequence or a protein sequence, what you can do is, by the analytical tools which are available in the bioinformatics, you can align them, okay, or you can keep the same sequence and compare the sequence with many sequences which are there already submitted in the database. So what will happen with this? When we submit our query data, what will happen is they will do an analysis in the whole database, and what they give is they give features like how much matching is there, how much mismatching is there, how much gap is available. Based on that, you can determine their function also. You can determine their structure, that is the 3D modeling, or you can do for the homology modeling. And based on that, you can also go for the phylogenetic tree analysis for understanding the evolutionary process in this. It includes, what are the things which is included in this is, there are two methods which are included. First is sequencing, that is the sequence assembly. Second is alignment, that is the searching. That is, it will search with a different analysis. So analytical tools basically helps us in this database. Like we are having tools like BLAST and FASTA. So the application, if you see the uses, the comparison of sequences in order to find the similarity, often to interfere if they are related or homologous. So if homologous sequences are there, like for example, if similar sequences are available, that they can give a score saying that they are very similar or they are nearby or in the same family or same type. That is what the homologous nature is. Identification of intrinsic features of the sequence, such as active sites, post-translational modification sites, gene structures, distribution of introns and exons if you are using eukaryotic samples. So all these things can be analyzed in the particular genome or genes sequence. Then identification of sequence differences and variations. So how much variations is there, how much differences is there, we can give a score for them, and we can give the deviation. That is, for example, X is the first generation. From that, the 10th generation, how much the difference is there in the evolutionary process, we can calculate and we can give the differences, such as the point mutation which has occurred, that is only one base has been changed, because of which the particular codon has been changed now. So or the single nucleotide polymorphism. So how much the polymorphism is present, differences are present in order to get the genetic marker. Basically, the study related to the SNP, that is the single nucleotide polymorphism, is very much useful for animal biotechnology as well as in plant biotechnology, and for analyzing for the drug discovery aspects, so that we know that how much the difference has occurred in their structure, in their morphological features by the mutational aspects. Then revealing the evolution and the genetic diversity of the sequences and the organism. So how much the similarity is there, how much the deviation is there, that we can identify. Like the example, you can say like horses and dogs. So you can say that how the evolutionary process has taken place, so that they have evolved themselves to a larger animal. Then identification of molecular structure from sequence alone. So you can take the structure, you can take the sequence, based on that you can create the molecular structure, and based on that you can analyze it. And genetic diseases, what are the different diseases are available for which we can act. See, for example, if you see the current site for the blast, you can find that it is having the sequences for the current diseases which are present. See, for example, tuberculosis, or it can be hepatitis, it can be chikungunya, it can be the SARS-CoV-2. So many things you can find it out in this particular database of the nucleotides, and from that you can take it and you can go for the alignments or the searching tool. So DNA sequencing. So in general, when we are doing the lab work or bench work, DNA sequencing plays a very vital role. So what we have to do, we have to follow different methods. Either we can go for the automated systems, or we can go for the manual systems for the gene sequencing or the DNA sequencing. So it is the process of determining the precise order of the nucleotides, or the order of 4 bases which are present, that is in which order they have been placed, like adenine, guanine, cytosine, and thymine in a strand of the DNA, how it is being placed, whether it is any deviations are there, that we have to identify. The second thing what we have to do is, it is expressed sequence tag, that is EST. So EST means it is a short subsequence of complementary DNA sequence. See, for example, if you have got your RNA, from the RNA, if you make a copy of that, or if you convert them, or you can say go for the reverse transcription process, if you take the cDNA, then what happens is that cDNA you have to make it into small fragments. In that small fragments, the short subsequences which are present, that you have to analyze. Then we are having the ESTs, may be used to identify the gene transcripts, and are instrumental in gene discovery and in gene sequence determination. So here it is very much important. So you can identify the gene transcripts, how it is present, and instrumental, that is the discovery also. That is, if it is a new one, that is new sequence also, you can identify. So if the closest match is nearly like 98%, then it is homology. If it is having a less than almost, you can say 92% or 91%, then they say that there are some new species are available, or new genus is available. We can identify using the tools based on the gene sequencing. So the methods which are used for the bench work basically are Sanger sequencing or chain termination methods, pyro sequencing, and shotgun sequencing methods. So this is a part of the fluorescent based sequencing chromatogram. So in this, if you see, you can find that after the DNA analysis is done, that is the DNA sequencing is done, that is the isolation of the DNA is being done, we will run it in the agarose gel. After running it in the agarose gel, we can take or particular DNA band which is available, that particular band can be taken up, and that we can go for the sequencing also, or you can give for the complete sequencing of the particular sample which you have got, and based on that you can go for the analysis. So in this, what happens is each is being tagged with a fluorescent dye. So what happens is when the primer is being applied for the sequencing, they will give a graph in which it is available. So then that graph is analyzed, and then we will get our that peaks, whatever is there, that will be analyzed, and that will be giving you the particular that is the DNA nucleotides in that. So protein sequencing. If you see the protein sequencing, it is a technique to determine the amino acid sequences of a protein as well as which conformation the protein adopts. The basic thing is adoption of the particular thing. Like if it is going to be 3D structure, it is going to be a simple structure, or it is going to be a secondary structure. That all things we have to determine here. And the extent to which it is complexed with any non-peptide molecules. That is, any other molecule other than this protein, if any glucose molecule, that is the carbohydrate molecule is there, or it's a lipid molecule is there, that we have to eliminate those things, and we have to take only the protein sequencing part here. So the methods which are used for the protein sequencing, you can say like Edmond degradation or mass spectroscopy. So if you see the mass spectroscopy, it is also called as liquid chromatography, mass spectrometry, that is being combined process. So here what we do is basically it is an analytical technique that ionizes the chemical species and sorts the ion based on their mass to charge ratio. So first thing what we have to do is, say for example, if you are taking a tissue sample, okay, so if you are growing the tissue sample and from that you want to extract the proteins. So what you can do is take the tissue sample, homogenize it nicely by the protein extraction process, isolate the protein. After isolating the protein, by using the SDS page, what we do is we run the gel. After running the gel, that particular band which is available, that will be cut and the particular protein will be eluted out. After the elution is being done, then what we do is we will take that whole thing and digest for the peptides, because we require the peptides. That is what are the different peptides are there. We are going to break the bond from the protein and make it into small amino acid sequences. That amino acid, what we do is, or the peptide mixture what we are having, that is passed through the liquid chromatography. In this liquid chromatography, if we charge our by electrospraying, by isolation, what happens is the peptide which comes out, that will be ionized peptide. So what happened now when we give it in the mass spectrometry? So mass spectrometry, what happens based on this, that is from the ground state, it goes to the excited state. So this, when the process takes place in the neutral gas levels, the collision actually takes place. So based on that, you will get a peak, or you can say like a band or graph, you can say, in which it is going up and down like that. Bands you will get it, or peaks you will get it. Based on that, that signal will be adopted and amplified, and that will give you as the graph model. That graph is then analyzed with the database, and that will give you that which amino acids are present and what is the peptide sequence which is present, and then they will be joined to give you the complete peptide sequencing here. So this is a manual method. When we are doing this manual method, this is a long process, because you cannot get the protein in a very easy manner, because you have to maintain the temperature. The temperature has to be maintained below 16 degrees. Then only the isolation can be done properly. In some cases, it has to be below 12 degrees also, because some will be very sensitive to the temperature. So the degradation can take place. So below 16 degrees, you have to maintain the temperature while working on this particular conditions. And in all the levels, it has to be maintained, that is below the level, so that it cannot go for the degradation process. So that has to be maintained here. So the sequence assembly, if you see regarding the bioinformatics aspect, sequence assembly means it refers to the reconstruction of the DNA sequences by aligning and merging the small DNA fragments. So that is what the process we are doing here. It is an integral part of the modern DNA sequencing method. So in this, what we are doing is long sequence is there that will be cut into small pieces. Then each fragment is being read properly. After reading the whole thing, we are reconstructing it. That we are again joining back the DNA by merging the information on various fragments. So the sequence alignment plays a very vital role here. Sequence alignment means it is a way of arranging the sequences of DNA or RNA or protein to identify regions of similarity that may be a consequence of functional, structural, or evolutionary relationship between the sequences. So if you are having a sequence, if you have made it into small fragments, each fragment either it can be overlapping or it can be a separate fragment. After that, we are going to have an analysis. That is, analysis means we will compare each sequences with the database. When we are comparing with the database, it can either go on right hand side or left hand side. So when the analysis takes place, it will verify with various alignment tools, that is the databases, and then it will analyze that whether the gaps are available, similarities are available, and then based on that, it will give us result in which we can go for the sequence alignment. So it can be done with both the DNA samples as well as in the protein samples. So it involves the identification of the correct location of deletion and the insertions that has occurred either of the two lineages since the divergence from the common ancestors. So in this, what we can identify, see for example, a new thing has been inserted, new fragment or new nucleotide has been inserted means we can identify that this is a new one and that is deviating. Then if it is deleted, that also we can identify by this particular sequence alignment process. So what will happen? Based on this, there are two types are there for the sequence alignment. So on the basis of the comparing sequence strand, it is of two types. First is called as the pairwise sequence alignment, and second is called as multiple sequence alignment. So if you see the pairwise sequence alignment, that is a pairwise sequence alignment only compares two sequences at a time. So two sequences means X and Y sequence are there. So it will compare only these two sequences. The next is optimality is based on score. So what happened based on the similarity, how much similar it is, that is how much similarity is there, based on that it will give a number that it is this much percentage similar or this is the value for this. Based on that score, we can consider that whether the similarity is present or some deviations are present now. Next is a pairwise sequence alignment consists of a series of paired bases, one base from each sequence. So now what we can do, we can take a pair of it. That is, we can take a subsequence of that and then we can compare that sequence also in this, so that we can identify that whether it is having a similarity with other organisms or not. So there are three methods are available, or three particular parts are available in the pairwise sequence alignment. That is, you can say like a global part or you can say local alignment. So if you are seeing the global alignment part, in that we can find that there are types of pairs are there. The first type in this is the matches. Matches means same nucleotide appears in both the sequences. The second type is called as mismatches. Mismatches means in upper one it is different, bottom sequence it is different. So that is called as mismatch. The other one which is called as that is the two different nucleotides are found in two different, that is the two sequences. And third one in this is called as gap. Gap means a base in one sequence and null base in the other sequence. Null base means there will be nothing, only a dash will be available, and bottom there will be a base sequence. So that when we are comparing, this is called as gap. So the algorithms are used here are Needleman-Wunsch algorithm, and the other one in this is Smith and Waterman algorithm, and the tool which is used in common is called as basic local alignment search tool, BLAST, which is very important for the protein aspects, but it can also be used for the nucleotides. So in the protein peptide sequence also this can be done. So if you see this particular sequences, you can find that the similarity is being provided with some colors. Okay, so the protein sequences are available. So similarities are available with most of them has been colored. If you see the certain one like here and here, here and here, these are actually the mismatches which are available. The maximum amount of similarity which is available, based on that the score will be provided, saying that this particular sequence what you have provided is very similar, almost you can say they are very closely related to the same family or same genus also. So multiple sequence alignment. So if you see the multiple sequence alignment, it is a sequence alignment of 3 or more biological sequences, generally protein, DNA or RNA. And the types which are used here is actually cluster W, um, probcons and muscle, which is used here. So this is how the multiple sequence alignment tool looks like, and when the comparison is being done. So you can see that they are comparing with 3 different types in this sequences they are comparing. So in this, they have taken from HBB human and MYG human. So there are 3 different types they have taken and they are comparing it. So when they are comparing, they are also finding the same thing, like they are having the gaps or not, they are having the mismatches, they are having the matches. So how it looks like, everything is analyzed here, and then based on that the score will be provided. Then databases, if you see, the biological databases are available, like they are the libraries of the life science information collected from the scientific experiments, published literature, high throughput experiment technology and computational analysis. So these all data, whatever is available, they have been put up in this databases. So there are different types of databases are available, like NCBI Unigen is there. So NCBI Unigen, that is, it is an NCBI, National Center for Bioinformatics database, that is of the transcriptome. Basically, whatever they have done, like whole genome project, or it can be single sequencing, 16S rRNA sequencing, or it can be a protein sequencing. So all the things have been submitted in this particular NCBI Unigen site. There is another one which is in collaboration. There is an international collaboration is also there. So in that, the DNA data bank of Japan is also very much important. So DDBJ, and European, there is a molecular biology laboratory. In that, the European Bioinformatics Institute plays a very vital role in this analysis. So all these 3 organizations, that is NCBI, DDBJ and the EMBL, will have all the common data, but only the format will be different. So in that only we can also find for protein specifically, that is SwissProt and TREMBL. So all these things actually gives us a work that in which we will understand that how the protein analysis or the DNA analysis can be, nucleic acid analysis can be done. So this is how the NCBI site is. In the NCBI site, either you can go with the DNA and RNA, or you can go with the all resources. In that all resources, you can take the search with all databases. In that databases, you can either choose the protein or nucleotide and then go for the particular search. So if you are clicking protein and you are giving the particular organism, in that a particular enzyme you can give. So what happens is the protein sequence or that particular enzyme you can get it, and then you can go for the particular searches, and you can find it out that, and you can compare it with other organisms, that whether any particular kind of that is the similarities are there or not. So I hope that in this particular session you have got an idea that how the bioinformatic tools, basically the web portals and the tools like BLAST and FASTA is actually helping for the drug discoveries, and how it is playing a vital role for the identification and 3D modeling. So in the coming sessions, we will see that how the 3D structuring using the bioinformatic tools plays a very vital role for the drug discovery. So thank you and have a good day.